Hi, today's topic is sex determination and sex-linked characteristics. We are going to explore the genetic basis of this and how sex determination occurs in different organisms, including ourselves. I hope you enjoy the lecture. Please think of questions that you will ask at question time. And here comes the voiceover. How sex is determined is one of the fundamental questions of biology. We have learned much on the subject. Model organisms such as the fly and the mouse have been crucial in furthering our understanding. When you look at this image, I want you to realize that although our sex is determined by our sex chromosomes, how we present our gender is largely cultural. Biological sex and gender are not quite the same thing. But this is not a gender studies class, so let's turn to the biology. Thomas Hunt Morgan is a pioneer of genetics. His influence on biology has been immense. He was born in Kentucky in 1866, the year Mendel published his paper. He set out to disprove the theory of inheritance in his fly lab at Columbia University. He demonstrated that genes are located on chromosomes. And he published The Mechanism of Mendelian Heredity in 1915, which was a very influential book. He established Drosophila melanogaster as a model organism to study genetics, and if you remember, Drosophila melanogaster is the fly. In 1933, he won the Nobel Prize for his chromosomal theory of genetics. He actually got the prize while he was at Caltech after he left Columbia. He was the first native-born American to be awarded the Nobel Prize in Medicine or Physiology. And his work relied on the painstaking work of finding spontaneous mutants, and these are very rare events. Morgan and his students developed the first genetic map of the fly. Here, this is a section of his book, his 1915 book, and you can see a map of 4 chromosomes, and he located the spontaneous mutations he discovered on these 4 different chromosomes. So how did Morgan get to learn so much about genetics and so much about flies? First, he began his studies of flies at the suggestion of a student. Morgan initially subjected the flies to all kinds of stress to select for mutants, but that didn't work very well. He tried to speed up evolution by breeding a particular trait for many generations, this trident in thorax, which is a coloration, if you look at the bottom, of the thorax, with 0 marking no coloration and 3 on the figure below marking the highest level of coloration. This trident coloration pattern on the thorax is a multigenic trait, so it's hard to study. But there was definitely hope for Morgan. So what did work for him? And that was the discovery of the white gene. And here is a white mutant fly taken by the excellent insect photographer Alex Wild. So he looked at thousands of flies. He was searching for a spontaneous mutation, which is a rare event. And then he found a white fly. Using this white fly, Morgan would go on to establish a modern theory of the gene, this chromosomal theory of inheritance, which is still very influential today. He also identified other spontaneous mutations, and this helped him realize that Drosophila has 4 pairs of chromosomes. And using all these different mutations, it allowed him to build this genetic map of Drosophila. Let's focus on the white mutant. Morgan's identification of this white-eyed male fly was incredibly important for his scientific career. And here I'm showing you what this white mutation looks like. So on the left, you have a wild-type fly that has a nice red eye, very clearly red on the left. And on the right, you have a white mutant that has lost all of the pigment in the eye. Using these mutants, he was able to do a lot of things. And this gave him the idea that sex determination works something like this, what's pictured here. So females had 2 X chromosomes, Males had one X chromosome, and he used the white gene as a sex-linked marker that allowed him to follow the segregation of the X chromosome. So let's see how he proved this. Here are the crosses Morgan did to determine that eye color was a sex-linked characteristic. So in the parental generation, the P generation, he crossed a red-eyed female with a white-eyed male. In the next generation, the F1s all have red eyes, both males and females. This is consistent with Mendel's first law, using dominant traits. And when he crossed these flies in the F2 generation, all the females were red-eyed and half the males were white. So from this, he inferred what the genotype of these animals were. He proposed that eye color was sex-linked. So now we're going to add some genotypic information to this cross. So you have your red-eyed female and your white-eyed male. The red-eyed female is wild type for the white gene in both of her X's. And the white-eyed male has one X, and that is a white mutant X and one Y. Both of these flies will make gametes. The female will only make one type of gametes, they're all wild type, whereas a white-eyed male will make a white X, which is a mutant X, and also give rise to a Y. The wild type X from the female and the mutant X from the male would produce red-eyed females who are carriers of the white mutation. All of the males will be red-eyed because the red-eyed female would contribute the X and the white-eyed male would contribute the Y. So males always contribute the Y to their male offspring. In the F2 generation, if you cross these F1 progeny, you will get a wild type X from the mother and also a mutant X coming from the mother. And from the male, you will get a wild type X and a Y. So this leads to the production of 1/2 red-eyed females, 1/4 red-eyed males, and 1/4 white-eyed males. Overall, 3/4 of the progeny is red-eyed. So now let's do the reciprocal cross, which was done to test Morgan's claim. So he crossed red-eyed female with white-eyed male, and he also crossed white-eyed female with red-eyed males. And in meiosis, you see the different gametes that can be produced. The red-eyed female in cross A at the left always produces a wild-type X chromosome. The white-eyed male can give either a mutant X chromosome or a Y. In the cross in B, the white-eyed female will always give a mutant X chromosome, and the male can give either a wild-type X chromosome or a Y. The normal separation of chromosomes results in half red-eyed females and half white-eyed males. In the F2 generation, if we cross the F1s, we can get both red-eyed females and white-eyed females. And this is consistent with the white gene residing on the X chromosome. So in this cross, we get 1/4 red-eyed females, 1/4 white-eyed females, 1/4 red-eyed males, and 1/4 white-eyed males. So with this data in hand, Morgan proposed this chromosomal basis of sex determination in flies. And here's an image from his 1915 book. XY sex determination also occurs in humans. And there are sex-linked characteristics that exist. Here I'm showing an image of a toothless, hairless man of Sind, which they were called in 1875. These people have a condition called anhydrotic ectodermal dysplasia. and it causes abnormality in their teeth and hair. So usually men have only 4 small and weak incisor teeth, and they only have 8 posterior molars. They have little hair on their body and become bald early in life. They suffer during hot weather from excessive dryness of the skin. So far, no instance has occurred of a daughter being affected, though daughters transmit the tendency to their sons. And no case has occurred of a son transmitting it to his sons. All of this evidence makes it clear that this particular genetic condition, anhydrotic ectodermal dysplasia, is a sex-linked characteristic. Another sex-linked characteristic is colorblindness. In colorblindness, usually men are affected, and they lose the ability to see red-green. So here on the left, you can see what people with regular vision see. And this is from the Ishihara test for colorblindness. Now, this is what red-green colorblind people see. And you see a loss of some of the numbers. So if you're colorblind, perhaps you just discovered this now, or you probably knew this already. But anyway, so this is a way to test whether you're colorblind. So how is colorblindness inherited in humans? Let's say a normal female mates with a colorblind male. During meiosis, the normal color vision female will produce gametes that contain 2 wild-type copies of the X chromosome. But the male will produce gametes that either contain a colorblind mutant version of the X chromosome or a Y. In the F1 generation, you should get all of their children with normal color vision. However, although the female will be a carrier of the mutant allele on one of her X chromosomes. If we do a reciprocal cross, where we take a colorblind female, and that would mean that both of her X chromosomes are mutant, and she mates with a normal color vision male, in the F1 generation, all of the daughters will have normal color vision but will be carriers. All of the males will be colorblind. So this is consistent with this particular condition being a X-linked trait. Things don't always occur as planned. And one example of this is nondisjunction. So let's say you cross red-eyed females and white-eyed males. All of the progeny should have red eyes, right? Based upon what we've discussed earlier in the lecture. But this is not always true. 1 out of 17,000 flies had white eyes. So this rare event was discovered by Calvin Bridges. And this is caused by nondisjunction. And let me give you a little more info about what I mean. In the P generation, if one of the parents was a white-eyed female and non-disjunction occurred during meiosis, she would produce a special kind of gamete that had 2 X chromosomes that were both mutant. And then the other gamete didn't have a sex chromosome, but of course had all the other autosomes. And she was mated with a red-eyed male that had a normal copy of the X chromosome, a wild-type copy. He would produce gametes either with an wild-type X or with a Y. And this is normal meiosis. Once fertilization occurs, in the F1 generation, you would get a red-eyed metafemale who would have 3 X chromosomes, and she would actually die. Then you would get a white-eyed female that had 2 X chromosomes that were mutant for the white locus and a Y. You would also get a red-eyed male who had no Y chromosome. And if you don't get any X chromosomes, they also die. So non-disjunction, in this case, results in white-eyed females and red-eyed males, which is totally different than what we saw before. This result is a very important result. It shows that non-disjunction can occur in chromosomes. And it also says something about how sex is determined in Drosophila, which is something that we will discuss later. So let's further explore non-disjunction. So it is a failure of a pair of chromosomes to separate properly during anaphase. This results in a cell with one extra chromosome, N plus 1, and a cell missing one chromosome, N minus 1. So these are the gametes that we're talking about. Non-disjunction is relatively common. It is estimated that 1 in 5 normal human pregnancies spontaneously aborts in the first 2 months because the fertilized egg had too many or too few chromosomes. A common error is the failure of a pair of chromosomes to separate during anaphase, resulting in a gamete with 24 or 22 chromosomes. Normally they should have 23. When fertilized by a normal gamete, you would get 47 or 45 chromosomes. So one will have 2n plus 1n, so it would become 3n in the embryo, and that's a trisomy, just for one chromosome, of course, not for all. And then in another case, you would have one embryo that lacks a chromosome, 2n minus 1, and that embryo would be 1n instead of 2n for one chromosome, and that's a monosomy. It's very rare for a successful pregnancy to result after non-disjunction, and it only works, you only have a chance with when you have the smallest chromosomes or sex chromosomes. Even then, it almost always leads to developmental abnormalities, some degree of mental retardation, and some examples are Down syndrome and Turner syndrome. So now, how many chromosomes do we have in the human genome? And how did we figure this out? So in 1912, von Winnewater counted 47 chromosomes. He thought females had 48 chromosomes as he could not see the Y, but this was not correct. In the 20s, Theophilus Painter saw 45 to 48 chromosomes in testicular tissues. He favored 46, but reported 48. In 1956, Johann Tigio and Albert Levin discovered a better chromosome preparation. This allowed them to clearly demonstrate the presence of 46 chromosomes, and their technique is used in karyotyping up until today. So here is the human karyotype. There are 23 pairs of chromosomes in the human genome, so there's a total of 46 chromosomes. The autosomes are highlighted here, and there's 22 pairs of those. And the sex chromosomes are highlighted in yellow, and the sex chromosomes here are X and Y. So this is a human karyotype of a male. So what else have we learned about chromosomes? Hermann Henking in 1891 noticed a peculiar structure in the nuclei of cells from male insects. Understanding neither its function nor its relation to sex, he called this structure the X body. Clarence McClung studied grasshoppers and recognized that it was a chromosome, this X body. Observed that the cells of female grasshoppers had one more chromosome than the cells of male grasshoppers. Concluded that accessory chromosomes played a role in sex determination. Eventually, it became known as the X chromosome. Nettie Stevens and Edmund Wilson demonstrated that females have two X chromosomes. They did this in insects. Males have a single X chromosome plus a smaller chromosome, which they called Y. X and Y chromosomes separate into different cells in sperm formation. And this happens through the process, of course, of meiosis. A sperm containing a Y chromosome unites with an X-bearing egg to produce an XY male, whereas a sperm containing an X chromosome unites with an X-bearing egg to produce an XX female. So this brings us to sexual reproduction in most eukaryotes. And this happens when a sperm fertilizes an egg, which is pictured here at the right. Sexual reproduction consists of two processes that lead to an alternation of haploid gametes and diploid cells. Meiosis produces the haploid gametes, and fertilization produces the diploid zygotes. And this is the cycle of reproduction in most eukaryotes. The sex chromosomes, as we've discussed, play an important role. The X and Y sex chromosomes are not homologous. Their gene sequences don't code for the same characteristics. Most genes on the X chromosome are different from the genes on the Y chromosome. In fact, the Y chromosome has only 5% similarity with the X chromosome. But they do pair during meiosis and segregate, which is what you're seeing here in this image below. As a consequence of this, males and females do not possess the same number of alleles at sex-linked loci. This difference in the number of sex-linked alleles produces the distinct pattern of inheritance in males and females. Now here is a look at what the sex chromosomes actually look like. So this is a human Y chromosome on the left and a human X chromosome on the right. So this cartoon of these chromosomes, you can see that the chromosomes have these different regions. The primary pseudo-autosomal region, which are highlighted in red, the secondary pseudo-autosomal regions, which are also highlighted in red, and these are at the tips of the chromosomes. And of course, they have chromosome arms and a centromere, as all eukaryotic chromosomes do. The X and Y chromosomes are homologous only at the pseudo-autosomal regions, which are essential for XY chromosome pairing during meiosis in the male. So how do we define sex? The mechanism by which sex is established is termed sex determination. There is monoecious, meaning one house species, organisms that bear both male and female reproductive structures, and these are hermaphrodites. And there's dioecious, meaning two houses species. So individuals in the species either have male or female reproductive structures. And the sex of an individual may be determined chromosomally, genetically, or environmentally. And we're going to explore all of those topics. So genes on the chromosomes are responsible for sex determination. But the inheritance of the sex chromosome does not guarantee the phenotype. XYs are usually a male in humans, but in rare instances they have a female phenotype, and this is caused by a recombination event that David Page will discuss in a video later on in this presentation. XX are usually a female. A few rare persons have male anatomy, but these people are genetically female. We refer to them as male because their sexual phenotype is male. There are several other systems to determine sex. XXXO, XXXY, which is what we've been focusing on, ZZZW, Haplodiploidy, environmental sex determination, and the Genic balance system. So we will go through these systems in more detail now. So for humans, as we've discussed, we have an XXXY sex determination system. These are our sex chromosomes. The smaller chromosome was called the Y, but it's not Y-shaped, so don't be confused by the name. Genes on the chromosome are responsible for sex determination. The non-sex chromosomes are called autosomes. As discussed, X and Y pair using the pseudo-autosomal regions. Genes found in these regions will display the same pattern of inheritance as that of genes located on autosomal chromosomes. And sex is determined by the presence of the male determining gene called the sex determining region, SRY, which is on the Y chromosome. Now this is true for humans and mammals, but not for all organisms with the XXXY sex determination system. For flies, it is different. but for humans, this is how it works. So how is sex inherited in humans? You have males and females. Males produce gametes that contain X or Y. Females only produce gametes that contain X. So therefore, the male determines the sex of the baby. If we look at the Punnett square for the F1 generation, we see a 1-to-1 sex ratio is produced. Now I encourage you to take a break. We're at the midpoint of the lecture. Pause, get a drink, stand up a minute. When you come back, we're going to discuss more about the Y chromosome in humans, some conditions that are caused by nondisjunction of sex chromosomes, and sex determination in some other organisms. Here's a cartoon of the Y chromosome. It has several regions, the PAR, SRY, some euchromatic regions, and euchromatic regions are regions that are transcriptionally active. It also has a heterochromatic regions that are transcriptionally silent usually. The PAR region, or pseudo-autosomal region, is critical for pairing and segregation during division. SRY is the sex-determining region on the Y chromosome. The NRY is a non-combining region, and it has 350 genes, including the SRY locus. And this is the region that does not recombine with the X chromosome. Here's another view of the Y chromosome. We have obtained experimental evidence that the SRY gene is required for sex determination in mammals. If you genetically engineer a copy of the SRY gene into XX mice, the mice, although sterile, will develop into anatomical males. The SRY gene encodes a protein that binds to DNA. This binding causes a sharp bend in the molecule. This alteration of DNA structure may affect the expression of other genes that control testes formation. Sex chromosomes in humans have many roles. Sex chromosomes contain genetic information essential for both sexes. At least one copy of an X chromosome is required for human development. The male determining gene is located on the Y chromosome, the SRY. A single copy of this chromosome, even in the presence of several X chromosomes, produces a male phenotype. The absence of the Y chromosome results in a female phenotype. Genes affecting fertility are located on both the X and Y chromosomes. A female usually needs at least 2 copies of the X chromosome to be fertile. Additional copies of the X chromosome may upset normal development in both males and females, producing physical and mental problems. Here's a summary of human sex chromosome phenotypes. Wild type, XX or XY. No X's, OO is lethal. XO leads to Turner syndrome, which happens in about 1 in 3,000 births, and the phenotype is female. Increasing the number of X's or Y's leads to Klinefelter syndrome. That happens in 1 in 1,000 births, and that leads to a male phenotype. Increasing the number of X's beyond 2 creates poly-X females, which occurs in 1 in 1,000 births. And there are XY females and XX males. In this HHMI video by Dr. David Page, who is an expert on sex determination and particularly an expert on the Y chromosome, he will discuss meiosis, sex determination, and some of the unusual outcomes that can occur.
Now what we're going to do is watch a video that connects 2 of this morning's themes, meiosis and sex determination. The question is, by what mechanism does an embryo come to carry a Y chromosome or a second X? The answer is found in meiosis, in the father. So in this video of human male meiosis, I want you to look for 2 things. Guess what they are? gene swapping, the swapping parts of paired chromosomes, and dividing by 2, halving the number of chromosomes per cell. So you'll remember that human cells have 23 pairs of chromosomes, but to illustrate the principles, we're just going to show 6 pairs here, 6 pairs of chromosomes, and we're going to look in detail at 2 pairs. We're going to look at the XY pair here and a pair of autosomes. And you see that we have it arranged so that mom's chromosomes, right? You got half your chromosomes from mom, half from dad. We have it arranged so that mom's chromosomes are on the left in red. The chromosomes you got from dad are in blue on the right. And what's the first thing that happens in meiosis? The very first step is you actually don't divide by 2. You multiply by 2. You double all the chromosomes, double all the genes. So at the first step in meiosis is you go to 4n, to a 4n stage. That's where we're beginning. Let's roll the video. Okay, so we're now going to blow up and focus on this pair of autosomes on the left. And you see there that recombination can occur at any point along the length of this autosomal pair. Let's see what happens when recombination occurs at a particular point. There's the swapping. We've just swapped a bunch of genes. The same can happen on the other arm of that pair of autosomes. Now we're going to turn over to the sex chromosomes. That's where the SRY gene is located on the Y. And it turns out that the X and the Y can recombine only within, they normally recombine only within their ends. Let's see how the swap occurs in detail. So we've just swapped parts of some stuff from mom and dad down at the other ends, like so. So at this point, how many copies of each gene, of each autosomal gene, does this cell have? 4. We still have 4. We have 2 copies of the X and 2 of the Y and 4 of every autosome. Okay. And what we're going to see is, what do we have to do now? We've got to go from 4 copies down to 1 per cell. So we're actually going to have 2 rounds of division. And you're going to see the X up here is going to go to 2 cells on the top. The Y is going to go to 2 cells on the bottom. We're going to get 4 sperm out of this 1 cell. Let's roll the video. Okay, here comes the first division. The cell on the top has 2 copies of every gene. The cell on the bottom, 2. Now comes the second division. Remember, we've got the X's in the 2 cells on the top, the Y's on the 2 cells on the bottom. And these are going to go on to mature into sperm. So we will now have X-bearing sperm on the top and Y-bearing sperm on the bottom. And now these sperm have to go in search of eggs. Let's roll the video. Now they're fired up. On the experiment on the top, the X-bearing sperm in purple are going to win. Down below the Y-bearing sperm, we end up with an XX fertilized egg, an XY fertilized egg. And then, as we said, the first 6 weeks of human development are anatomically, histologically indistinguishable in male and female. We progress up to the 7-week stage right here, and it's only at this point that the SRY gene fires and leads to the birth of a male or female. Okay, so how do XX males carrying SRY arise? How do XY females deleted for SRY arise? I want you to remember that the XX males are carrying a terminal portion of the Y chromosome. The XY females are missing a terminal portion of the Y chromosome. We're going to see how that happens in a rerun of a part of this video we just watched. Let's run that video now. Okay, so here again we got the 6 chromosomes that are representing our 23 pairs. Now we're going to look just at the XY pair. And we're going to watch an aberrant recombination or gene swapping. So there again is the SRY gene on the Y chromosome. Recombination would normally be restricted out at the very ends of the X and the Y. But occasionally, an aberrantly placed recombination event. Look, that one is too far down. And look what's happening. The SRY gene is being passed over to an X chromosome. And look, here's a Y chromosome that has lost SRY as a result of this misplaced swapping event. So we end up with an X chromosome that is SRY plus and a Y chromosome that is SRY minus. That's exactly how XX males and XY females in humans are generated.
A normal individual has 46 chromosomes, and this includes an X and Y if it's a male. In Turner syndrome, the Y chromosome is missing, so they have 45 chromosomes, and including just one X. This affects 60,000 girls and women in the United States. The disorder occurs in 1 in 2,000 births, with about 800 new cases diagnosed each year. The phenotypic effect is mild because their cells have a single functioning X chromosome like those of XX females. But they do not develop secondary sex characteristics such as breast tissue and underarm and pubic hair. People with Klinefelter syndrome have 47 chromosomes. They have an extra X, so that is 2 X's and a Y. And you can see someone with Klinefelter syndrome pictured on the right. It happens in 1 in 500 to 1 in 1,000 live births. The phenotype includes tall stature, infertility, gynecomastia, which is excessive development of the male mammary gland, and hypogonadism, which is characterized by decreased size and activity of the gonads. In XYY syndrome, which is a human male with an extra Y chromosome, that male has 47 chromosomes. The 2 Ys are highlighted here in green. This is a fairly common condition, with 1 in every 851 male newborns is an XYY boy. Some studies have suggested lower IQs, increased aggression, and reduced fertility in XYY males. The only really significant difference that I think is anywhere near scientifically valid is that they tend to be taller and usually greater than 6 feet tall. Most of the time, this condition is undetected in childhood and even in adulthood. The phenotype is very poorly understood. I think there's a lot of rumors about how XYY males behave and things like that, but I don't think there's a lot of science to support them. Multiple X chromosomes can also be inherited. This leads to poly-X females. It's a relatively common cause of learning difficulties, particularly language-based disabilities. Dyslexia is an example in females. Mental retardation rarely occurs, so intelligence overall is normal, but the severity increases with the number of X's beyond 3. Infants and children with Triplo X syndrome may tend to have delayed acquisition of certain motor skills and delayed language and speech development. Affected females are often of tall stature, and although sexual development and fertility are usually normal, some may have delayed puberty and/or fertility problems. How can an individual inherit 2Y or 2X chromosomes? During anaphase 1 of meiosis, the chromosomes with 2 chromatids normally separate. During anaphase 2 of meiosis, sister chromatids separate, each chromatid becoming a distinct chromosome. But if this process is disturbed and there is nondisjunction, then a homologous pair does not separate in meiosis 1, so this could be one way that nondisjunction would occur, or a sister chromatid does not separate in meiosis 2. Now we're going to move on to sex determination in other organisms. Here is a chicken. And birds have a different way of determining sex. They use the ZZ-ZW system. In this system, females are heterogametic, ZW, and males are homogametic, ZZ. The inheritance of Z-linked characteristics in females is analogous to the X-linked characteristics in male mammals. This happens in birds, moths, some amphibians, and fishes. Another system for sex determination is the XXXO system. This system is the simplest system. It is found in grasshoppers and in other organisms. Females with two X chromosomes, XX, they are the homogametic sex. And males have only one X chromosome, no Y, and they are the heterogametic sex. Sex determination can also occur when there are no sex chromosomes. One example is Haplodiploidy, and this occurs in bees, wasps, and ants. Genic sex determination occurs in plants and protozoans, and environmental sex determination happens in marine mollusks. In genic sex determination, this happens in organisms that lack sex chromosomes. Both sexes have the same number of chromosomes. Here we have a plant, a flower of a plant. But genes on the chromosome sets determine the sex. Sex determination is happening in autosomes. Here's a bee. Bees are an example of a species that use haplodiploidy for sex determination. Haplodiploidy occurs in organisms that lack sex chromosomes. Examples are bees, wasps, and ants. It's determined by the number of chromosome sets. Males are haploid and females are diploid, as you can see here on the right. Males develop from unfertilized eggs. Females develop from fertilized eggs. Males produce sperm by mitosis, and all offspring have the same paternal genes. Environmental sex determination happens in a variety of animals, including reptiles, like the one pictured here. It happens in organisms that lack sex chromosomes. Another example is the marine mollusk. They start out as larvae, and over time, they settle on a solid, unoccupied substrate, okay? And then they develop into a female. This attracts other larvae that settle on top and develop into males, which become mates for the original female. After a while, the top male develops into females, attracts larvae who develop into a male. This is called sequential hermaphroditism. Basically, sex is determined by the position on the stack. Some organisms use a genic balance system to determine sex, and this is true of the fly. The ratio of sex chromosomes to autosomes determines sex in the fly. Genes for femaleness are on the X chromosome. Genes for maleness are in the autosomes. The ratio of the sex chromosome to the number of autosomes, this X to A ratio, determines sex. Here you can see the 3 autosomes and the 2 sex chromosomes. There are 4 sets of chromosomes. 2n equals 8. That includes 3 pairs of autosomes, and they make 1 set, and 1 pair of sex chromosomes. So when the XX to autosome ratio is 1, then you get a female. So that AA means 1 set of autosomes. When the XY to autosome ratio is 0.5, you get a male. If you have an XO to AA ratio of 0.5, you also get a male. If you have an XXY to AA chromosome ratio, you get a female. Here is a table of the chromosomal complements and the sexual phenotypes that you would get in Drosophila. If the ratio is less than 0.5, you get a metamale. And if the ratio is greater then you get a metafemale. Metamales are weak and sterile, and metafemales have developmental problems. So it's important in Drosophila to get the X to autosome ratio right. So that brings us to the subject of dosage compensation. Females have more X chromosome than males, and this is true in Drosophila and in mammals. Does that mean they have twice the expression of genes on the X? Just now we learned a little bit about how flies handle this situation. How do mammals do it? So Mary Lyon in 1961 had a hypothesis that the Barr body, which is this area pointed out in the nucleus of a mammalian cell, this little dark region at the left that is being pointed out by the arrow, she had a hypothesis about what these Barr bodies were. She believed that only one of the two X chromosomes is genetically active in female cells. And her work showed that X inactivation occurs around the 16th day of embryonic development. What can affect the number of Barr bodies? It is simply the number of X chromosomes that you have. So if you have one X chromosome, as in the upper left, you get no Barr bodies. If you have two X chromosomes, as in the upper right, you get one Barr body. If you have three X chromosomes, which is on the lower left, you get two Barr bodies. And if you have four X chromosomes, as in the lower right, you get three Barr bodies. So in all of these situations, only one X chromosome is active. These Barr bodies are physical evidence of X inactivation and allow for the proper dosage of X-linked gene expression in mammals. So let's review dosage compensation for X-linked genes. In mammals, one of the X chromosomes in somatic cells of females becomes inactivated. This is the lionization process. Inactive chromosomes can be seen as Barr bodies inside the nucleus. This very condensed chromosome is transcriptionally inactive. X chromosome inactivation is random and is independent from cell to cell. X inactivation in humans occurs at the 500 to 1000 cell stage of embryonic development. However, once inactivated, all descendant cells will inherit the same inactivation pattern. So once a cell inactivates the X chromosome, all of its daughters will have the same chromosome inactivated. In Drosophila and C. elegans, they have an X chromosome autosome balance system. Sex is determined by the ratio of the number of X chromosomes to the number of sets of autosomes. In Drosophila, a 1-to-1 ratio gives females, and a 0.5 to 1 gives males. Y has no effect on sex, but is required for fertility in males. The X chromosome also displays dosage compensation. This involves higher transcription rates of X-linked genes in males. So there's no X chromosome inactivation in flies. In C. elegans, individuals are either male XO or hermaphroditic XX. Dosage compensation involves a reduction of the transcription of genes in the female X chromosomes by half. So in C. elegans, you don't ramp up gene expression on the X chromosome in the male. You instead achieve equilibrium by reducing the level of expression of the X chromosomes in the female by half. So now I want to review some genetic terms that were discussed in the lecture today. And here we go. Autosomes, chromosomes not represented differently in the two sexes. Sex chromosomes, chromosomes that are represented differently in the two sexes. A heterogametic sex produces gametes with two types of sex chromosomes. A homogametic sex produces only a gamete with one type of sex chromosome. Nondisjunction, abnormal chromosome segregation during meiosis. Aneuploidy, the number of chromosomes deviates from the norm. Euchromatin, this comprises the most transcriptionally active portion of a chromosome. Heterochromatin, these are regions of the chromosome that are modified to prevent transcription. Wild-type allele, a functional allele of a gene. Mutant allele, an altered allele of a gene that changes the gene's function. It usually reduces the gene's function or eliminates it. Well, I hope you've enjoyed today's lecture. Next time we will explore some extensions and modifications of Mendel's principles. See you next time.